Hello, everyone, and welcome to this first edition of In Fact. In Fact is a YouTube channel who try to explain in a nutshell what are the cutting edge research in life science. I'm Elliot MacDonald, and I have an M.Pharm degree. So today I will present you the topic of creating new neurons in the adult brain. This is the first part of this topic. I will give you an overview of the current state of research in creating new neurons, but first, I will introduce what are neurons and glial cells and how do they assemble to form the brain. Then, is it possible to lose neurons? And can we have new ones? And why is research in generating new neurons so important? So first of all, what are neurons? Well, neurons are electrically excitable cells which transmit signals through the body. So here's a picture which simplifies what a neuron is. So on the top there, you got dendrites. Dendrites are the part of a neuron which receives information. In the middle there, you got the cell body with a nucleus in black here, and down there you got the axon. The axon is a part of the neuron which sends the information to another neuron via a process which we call a synapse. Here. How does that work? Well, let's say you have a signal which starts here. Let's follow it. You can see that the signals actually goes through here and then goes by the synapse and into the neuron. To make sure that the signal flows properly through the axon, there is what we call a myelin sheath, which is around it. It's a sort of tissue, an isolation, which makes sure that the electrical signal is well conducted and well transmitted. Neurons are involved in many roles. They are involved in movement control, in senses, memory. There's a lot of different roles for neurons. In this video here, I'm going to show you actually how signal is transmitted in reality. And so you will normally see white flashes, which will normally start from the dendrites, go through the cell body, and then to the axon. Watch closely. So you can see in this frozen picture there that you have your dendrites, which are lit up, your cell body, which is lit up, and then your axon there, which starts to transmit to another cell. Okay, so now we have saw what neurons are, but how do they organize themselves in the brain? So, in the brain, you got two matters, the gray matter, which is here, in gray, and the white matter, which is in white here. In the gray matter, you found the cell bodies here of the neurons. Whereas in the white matter, you found the axons. So in the gray matter, you have neurons, so the cell body of the neurons, and the axons are in the white matter, and the white matter is the place where communication between neurons is made. So, is the brain only made of neurons? Well, neurons are not the only cells in our brain. There are other cells called glial cells. In Greek, glia means glue. There's 3 types of glial cells and 3 times more glial cells than neurons. These cells are essential for the support and the correct function of neurons. You have, as I said, 3 types of glial cells. You have the astrocytes, which are called like that because they look like stars. You have oligodendrocytes, which are involved in wrapping myelin, where this was before, which is isolating the axons. And you got microglia, which is involved in immune response. Astrocytes have many roles. They're actually one of the most important cells, most present cells in the brain, and they're involved in support of neurons, but also for their function. They're giving nutrients, resources, anything that the neurons need to properly function. And also, quite interestingly, astrocytes are involved in neurogenesis. So the fact of creating new neurons. We'll see later how. And okay, so we got neurons from one side and glial cells from another. And how do they actually work? Well, all of them are working like a sort of family all together. You have astrocytes, as you can see on the picture in green, which is closely linked to neurons and have some, actually touches the neurons. And you have oligodendrocytes, which are wrapping themselves around neurons to make sure that the signal is properly transmitted through the axon. And you got the microglial cells around here. So you can see that all of them are sort of forming like a cluster and walking all of them together. Can we lose neurons? Yes, we can, and in different cases. During a traumatic brain injury, for example. Here, for example, you can see this black spot, which means that neurons were lost. When we have a stroke or VCA, a stroke happens actually normally in elderly patients, and it's caused by a lack of oxygenation in the brain. What does that mean? It means that basically blood in certain part of the brain, blood stops to circulate. And if blood stops to circulate, well, neurons don't have any more oxygen or nutrients, and they just die. Here you can see an important spot, which means that a lot of neurons were lost. You can lose neurons in case of if you have a neurodegenerative disease, for example, Alzheimer, Parkinson, and so on. What is the neurodegenerative disease? A neurodegenerative disease is a brain disease where you have an abnormal loss of neurons. That means that you're losing neurons at a quicker rate or a quicker pace than normally you should. And for example, here you have a normal brain, which looks which looks fine, and you have an Alzheimer brain on the right, where you can see that you have a thinner white matter and thinner grey matter, and you can see that you have bigger ventricles. So you can guess that there's been a lot of neurons which have been lost here. In aging, of course, is natural. You lose neurons with age, and this is young adults which have a normal brain, and you have here an old adult which has no diseases whatsoever, and you can see that you have a reduced grey matter and white matter, but mostly a reduced grey matter. So it has obviously this patient, he or she has lost neurons during ages, but this is a natural cause. Can we have new neurons when we are adult? Well, for a long time, it was thought that the brain could not regenerate new neurons. It's not entirely true. There are some places in the brain where a process called neurogenesis still occurs. Neurogenesis is a process which creates neurons. There's two places in the brain where this happens. You have a subventricular zone, which is under the ventricle here, and you got the dentate gyrus subcranial zone, which is in the hippocampus, which is here. Both of them contribute to create new cells, new neurons, which will replace neurons in the olfactory bulb. The olfactory bulb is involved in the sense of smell. Specifically, neurons also in the dentate gyrus, which are here, contribute to the creation of new memories as well. Okay, so we have saw that we can have and we can actually lose neurons and we can have new neurons. But why is there so much research on creating new neurons? Well, natural neurogenesis is not always adapted and sufficient to replace lost neurons in the brain. Indeed, mature neurons as well are unable to multiply themselves and replace lost neurons. So what does that mean? Well, that means that when you have a traumatic brain injury, or for example, a neurodegenerative disease or anything which causes a loss of neurons, well, you can't have them back. They're not going to grow back anymore. So when these neurons are lost, they're lost forever. So there's an important medical need in learning loss because there is no treatment yet to create new neurons in the adult brain. In neurodegenerative diseases, for example, as Parkinson or Alzheimer, it is not possible yet to regenerate new neurons. There is no treatment available. And this approach of creating new neurons in the brain is very interesting because it would solve these problems in these diseases. Indeed, by creating new neurons, we could restore lost functions. And what are the strategies to generate new neurons? Well, if you want to know more, and if you want to have more neurons, subscribe and you will see this in the second part of this topic. Thank you very much for your attention. Hope you enjoyed the video and see you at the second part.